Mpeg-G. Background: the advent of high-throughput sequencing HTS technologies has revolutionized the field of quantitative biology. Availability of large collections of genomic information has now entered everyday practice and has become a cornerstone of a number of disciplines, ranging from biological research to personalized medicine in the clinic. At the moment, genomic information is mostly exchanged through a variety of data formats, such as FASTA/FAST for unaligned sequencing reads and SAM/BAM/CRAM. The ISO/IEC 23092 Mpeg-G standard aims to provide a unified format for the efficient representation and compression of such diverse data, both for file storage and data transport. In order to do that, the standard is divided in several parts. Structure of the standard. The Mpeg-G standard utilizes technology and data representation architectures previously validated in the field of digital media. They allow to compress and transport genome sequencing data even in complex scenarios, for instance when access is needed to large amounts of possibly distributed data, or when part of the data needs to be encrypted for privacy reasons. Conceptually, such requirements lead to the definition of a number of mutually interrelated mechanisms, which are summarized in the following list: data format and compression, data streaming, compressed file concatenation, incremental update of sequencing data and metadata, selective access to compressed data, e.g. fast queries by genomic range, metadata association, enforcement of privacy rules, see rules, selective encryption of data and metadata, annotation and linkage of genomic segments. In turn, some of these topics have been collected together in order to make the standard easier to understand and implement. As a result, the ISO/IEC 23092 standard is physically structured as a series of separate documents as follows: ISO/IEC 23092-1 Mpeg-G Part 1. ISO/IEC 23092-1 specifies how the genomic data is organized within Mpeg-G structures for transport, i.e. streaming and storage. Formats of genomic record, reference record, Mpeg-G file, and transport stream are defined in this part. It introduces access unit as the container of the compressed genomic data and provides a reference conversion process among different formats. ISO/IEC 23092-2 Mpeg-G Part 2. ISO/IEC 23092-2 specifies the syntax and methods for Mpeg-G lossless compression of sequencing data and lossy compression of associated quality scores. Mpeg-G, as is typical for Mpeg standards, only specifies the decoding process, while the encoding process is left open to algorithmic and implementation-specific innovations. All Mpeg-G conformed decoders produce identical outputs from the multiplex bit streams included in Mpeg-G files and the data streams in streaming scenarios. The input data of the encoder are genomic records or metadata, with optional reference data, while its output is Mpeg-G file or transport streams. ISO/IEC 23092-3 Mpeg-G Part. ISO/IEC 23092-3 specifies a metadata format and provides genomic data representation API. Part 3 specifies how an Mpeg-G compliant bitstream can be integrated with metadata, as well as mechanisms to implement access control, integrity verification, authentication, and authorization mechanisms. This part also contains an informative section devoted to the mapping between SAM and Mpeg-G data structures, including backward compatibility with existing SAM content. It defines ISO/IEC 23092-4 Mpeg-G Part 4. ISO/IEC 23092-4 specifies genomic information representation reference software, referred to as the genomic model (GM). It consists of two components, the reference encoder software and the reference decoder software. While the reference decoder software is provided to assess the conformance to the requirements of ISO/IEC 23092-1, ISO/IEC 23092-2, and ISO/IEC 23092-6. The reference encoder software serves as a guide for the implementation of the aforementioned. The reference encoder software called Genie is an open-source software developed by a group of individuals from multiple universities and companies around the world. It features the following components: ISO/IEC 23092-5 Mpeg-G Part 5, ISO/IEC 23092-5 specifies conformance of the coding of genomic information. Part 5 provides a means to test and validate the correct implementation of the Mpeg-G technology in different devices and applications to ensure the interoperability among all systems. It specifies a normative procedure to assess conformity to the standard on an exhaustive set of compressed data. MIMEType and Filename extensions. No MIMEType RFC 6838 based INA media type currently defined for Mpeg-G file. No conventional file extensions are defined.